Gosh. Well, you know, what I typically do if I'm going to give a talk, I try to prep it way ahead of time, and then I just kind of let it mull through my head and make tweaks and changes. Exactly. So hopefully it won't be too rough, because I was kind of counting on this one in August.
Mic check 1, 2. Give me a thumbs up when you're good. Okay. Yes, sir.
So, we've got 2 slideshows here.
Okay.
This one is for Melissa.
Okay.
The myths and mounds.
Yeah.
Rhyme that with M.
questions, please wait till we get to you with the microphone.
Yeah, perfect. You already thought of it. It's like you've done this before.
I have. Good afternoon. Welcome to the monthly meeting of the Arkansas Society of Free Thinkers. Happy solstice to all those who celebrate. And if you don't, why not? I understand that a coven that's loosely associated with our group is going to attempt to sage the state capitol grounds at midnight tonight. I'm Bern Bradley. I'm one of the organizers of Free Thinkers. We are a nonprofit organization devoted to maintaining the separation between religion and government, advocating a scientific and humanistic viewpoint, and improving society. Find out more about us on the web at arfreethinkers.org, or, and the most reliable way, is by getting to know some of the members and organizers by coming to our events. Our calendar is on meetup.com. Just search for the Arkansas Society of Free Thinkers. Opinions expressed are not necessarily those of the Arkansas Society of Free Thinkers and belong solely to the speaker. Having said that, here with the free thought of the day is Brian Shank.
Good afternoon. Well, as Bern pointed out, it's winter solstice. It's a summer solstice today. So that's June 21st. Or more precisely, in Little Rock, the solstice was at 3:24 a.m., the point at which the Earth's tilt toward the sun is the greatest. It's at that moment this morning at 3:24 a.m. that happened. So we have more daylight hours today than, I suppose, any other day of the year. So it's about 14 hours and 30 minutes is from my research on this. And the winter solstice is 9 hours and 50 minutes, so that's about 4 hours and 40 minutes difference. So I have two quotes I was going to list today for the occasion. One way of celebrating the solstice is to consider it a sacred time of reflection, release, restoration, and renewal. That's a quote from Sarah Van Redonk. Another quote was interesting. From the Lifestyle and Wellness website, Will the Moon, there is an inspirational essay published there, Notes from the Universe section titled The Longest Day Doesn't Want Anything from You. So the world will want more from you on 22nd June. It always does, but on the 21st, the sun lingers like it has nowhere else to be. The longest day doesn't ask more from you, it simply gives you more. Thank you.
Here with this month's member story is Chris Barecki.
Greetings. Happy summer solstice and happy Father's Day. Member story is a section where we sort of get to know each other, share our experiences in life, share some of our perspectives on those experiences, and it is a story. Today I was going to share with you sort of a merging of two stories and the synthesis that I gained from those two stories. And I'll start with this. Last weekend we participated in Soma Pride. And I tell you, it was a joyous experience. There were so many thousands of people there that it was such a big deal. The city infrastructure, everything was cooperating towards this production of joy, this production of culture, this celebration of a new set of values that if you had gone back 30 or 40 years and told somebody that this would be happening in the streets of a place like Little Rock, Arkansas, they would think you were absolutely nuts, right? Think about what the mentality was like just a generation ago. And think again about how the organizers of Pride have built something out of pure positivity, because Pride's not about criticizing anyone else. It's not about tearing anything else down. It's not about complaining. It's not about a gripe session. It's about the production of joy for a day. It is about, bringing people together who want to come together but live in a world that tends to tear us apart. And so it is the rejection of that tearing apart and bringing together allies and friends and really anybody. And it's very inclusive, and it is a new direction for the world to go. They've created that in a place where people would say that's impossible. As you think about your own beliefs about what is possible and what is impossible here in the center of Arkansas, think about what the people achieved in real life within your living memory. Think about how positivity can happen, how positivity can draw people in, how positivity and creating something new out of the old can overcome a lot of adversity and a lot of resistance and become the most joyous event on the calendar. And now there's not just one Pride. Now there's a calendar of events. There's multiple events all over Arkansas if you go to the Arkansas Pride website and see what they have produced. And then that makes me think, what can we as secular people produce in a similarly joyful way? What kind of organization, what kind of new culture can we build? Starting right here, starting from scratch, just like the Pride people did, just like the organizers in the 90s did when they first started conceptualizing something for Arkansas. Where can we be 30 years from now? And whether you're planning to be there or not, doesn't matter. What can you be a part of that is still here and much, much bigger and a big chunk of the culture in a town? Think along those lines. Think big, because that is where the future lies. It's among the people who think big and who work with the building blocks of positivity. The other part of my synthesis, I met up with a friend of mine, demographically very similar to me, but according to our lovely little cell phones, we live in different worlds. My friend gets most news from x.com, a little from Instagram, YouTube videos, probably a dash of Fox News in there. I'm just judging by what this friend, shares with me that they think is funny and whatnot. And this friend and I were, hanging out, and the topic of the war in the Strait of Hormuz came up. And I had the opinion that, there's going to be some geopolitical consequences to the United States losing that war. He said, what are you talking about? The United States didn't lose that war. We blew the hell out of them. I was like, really? Because, that tells you where the information is coming from, and it's different. We are now in such an Orwellian world. This is straight out of 1984, where some people think we won the war and some people think we lost the war. And I can't tell you if I'm right and he's wrong. I could have gotten into an argument about, no, according to all my internet sources, we lost the war badly, and we were kicked out of the Persian Gulf, basically, and Iran runs the show, and we had to capitulate, and we're going to send them $300 billion to please reopen the Straits of Hormuz, and we're so sorry we ever fought you. His perspective is completely different. We blew the hell out of them. They are in full retreat, and we're calling the shots now. Like, what is the truth? I don't know. We could look at objective reality and say that, gasoline prices fell a little bit, so that's inconsistent with my point of view that, the straits are probably still mostly shut down. Or we could, look at other pieces of information, but why are we even in this situation in the first place? Why are we arguing about something as big as whether our own country won or lost a war? That's the internet for you. That's what the internet can do for us. We don't know the basic things that are going on around us because the algorithms deliver different information to different people based on what they linger over or engage with or the other things they've liked and watched. So The internet, in this sense, was a dead end because I could have argued with my friend all night long and we would just be getting our strings pulled by the algorithms and put into a fight in real life about who's right and who's wrong and who's dumb and who's smart. No, it's just we were fed different pieces of information. That's all that the conflict is about. So why argue with that? The synthesis with pride is that creating something in reality is much more rewarding than living your life on the internet. There's a study about how people who watch more Fox News are less informed about the world than people who watch, no Fox News. What if that has been extended now to multiple platforms, to all the algorithmic platforms, and the more you absorb, the less well-informed you are? Bigger consequence, the more you absorb, the more likely you are to be in conflict with the people around you who get a different narrative fed to them. And then suddenly you don't have a life in the real world anymore because you've alienated all your friends and family. Make something in the real world. Pride is the example. Us arguing about whether the war was won or lost based on internet information is not the way forward. And so that's my synthesis of two very unrelated experiences over the past week. Thanks, y'all. Okay.
If you drop the script, you must pick it up. Today's skeptical sidebar is done by Melissa Ray, and I'm going to very quickly try to pull. There we go. We are working. And now, please welcome, with a stack of books, Melissa Ray.
Okay. I promise I'm not going to read all those to you. Hello. Let's see. Presenter, sure. Oh, that's not what I want. Sorry. Is that what I want? Kind of. We'll go with that. Hi, my name's Melissa Ray, and I'm here to talk about myths and mounds. I'm here because Mason and Kristen, big-time ASF members here, are my friends of many, many years, and I've told them some pretty bizarre, crazy stories through 5 of those years of our friendship. For 5 years, I worked at Native American archaeological mound sites throughout Arkansas. So if you've heard of Toltec mounds, known as Plum Bayou mounds now, or Parkin, you might have heard of them. That's a mound site up in northeast Arkansas where DeSoto actually visited. I've worked at both of those mound sites. I'm also certified in professional archaeological excavation. I've done mound site digs, and I got a bachelor's degree in history, so I hope I know what I'm talking about, but if you don't believe me, believe in archaeologists because they're even more well-versed than I am. So I always tell people to go get the expert opinion. Now, if you've heard of a mound before, you're probably thinking burial mound, and that's okay. Pretty much every person that came into one of our parks, one of our mound sites that I worked at, thought that all of the mounds that I worked with were burial mounds. That's not the case. There's actually also pictured, that's not a burial mound, fun fact. There are 3 main types of mounds. So the one that you'll most often see, not really see at mound sites, are ceremonial. They're very low to the ground. They're like 8 to 10 feet tall or lower. So in the 1800s, thousands of years after these mounds were built, 1,000 years after the mounds were built, farmers would come through and plow the land. They don't like hilly land. They don't like mounds. They like flat land, right? So they're going to plow through all these mounds. Ceremonial mounds are low to the ground so that people can get on top of those mounds and gather and have ceremonies, though. So 15 of the 18 mounds at Toltec were ceremonial. So we talked about solstice today. One of the ceremonial uses of these mounds was a calendar to tell when the solstice is. So if you go to this little right-hand side ceremonial mound that you kind of can see, that used to be Mound H, and you used to sit on top of that mound 1,000 years ago and be able to tell when the sun set on other mounds when it was during the year. So it acted as kind of like a Stonehenge almost. There's other ceremonial uses, any kind of parties that you have today in modern times. Think of back then when Native Americans were gathering here. They would have parties with big, feasts and play games and play musical instruments on top of these ceremonial mounds. That's most of the mounds out there. So if your granddad comes in to the park I used to work at and say, I found a mound on my property, probably not a burial mound, probably not even a mound, but if it is a mound, it's probably going to be a ceremonial mound. Then there's platform mounds. They're very tall, they're high. These were 50 feet, 39 feet tall here at Toltec. They are just so that a chief or a priest or some kind of leader can live on top of that mound. And then there's the burial mound. There's just one of 18. This is indicative across the southeastern United States. Any mound site that you're going to talk about, there's probably just a few burial mounds at that site. So that is the first myth that I got when I was working in parks at these mound sites. You're probably wondering who built these mounds. A lot of people think giants. I don't know why. Okay, we got so many people come into the visitor center and they come up to the front desk and they say, I read everything in the museum. I spent 10, 15 minutes in there, but what about those giants though? Native Americans built these mounds. 3 reasons why this myth of giants building these mounds are so prevalent even today is this first picture. It's deep-seated racism is the first reason. And this first picture is a news article back in the 18th century where Indians, or Native Americans in this case, but it's called an Indian on here, they're labeled as a different race entirely to the mound builder. So mound builder is on here as well as a different race. There was a 18th century widespread racism towards Native Americans. This was the time right before American Indian removal. People did not think, your average farmer Joe and sometimes your archaeologists did not think that Native Americans were advanced or civilized enough to have engineered and built mounds that high. One of the reasons is sometimes mounds are dug into nowadays and we can't even put them back together the way that originally they were. These were engineering marvels, okay? Just a pile of dirt, one basket load of soil at a time can be an engineering marvel, and they just didn't believe that Native Americans could do that. They didn't believe that their artifacts were, anything like that. So the first reason is deep-seated racism at the time. So obviously it was an ancient white race, right, of giants from the Bible, scripture, whatever they pulled from that built the mounds. There was also money making, right? It's all about money. So the second article here, there were thousands of these articles put out in the 18th century and early 19th century of giant skeletons. And you still hear about these things today. So you see in the third picture a skeletal remains photo. And half the time, well, actually probably 90% of the time, that giant skeletons are a thing in the media even now, it's probably a mastodon or a woolly mammoth or something that they have accidentally prescribed as human or giant remains. Instead, they just didn't know about any kind of Pleistocene era, extinct mammal. Especially if you're Farmer Joe in the 1800s and you find something on your property, you're gonna see dollar signs immediately in your eyes, right? You're gonna think, I can make money off this if I call it a giant compared to what it actually is. You're gonna think, I'm gonna go to the local newspaper, the newspaper's gonna make money, we're all gonna make money, some schmuck is gonna come buy all these artifacts on the black market and we're gonna make some money. So that is why this prevails, and YouTube. This was a quick Google search that I did like an hour before this, and this is just a screenshot of the hundreds of YouTube videos that are on the internet right now about giants building these mounds. I don't know why it's still so popular, but the only thing I can think of is we're not really taught in schools about mounds very much, and you might not have even known that we had mound sites here. So a more interesting version of the truth is that giants built these mounds instead of just Native Americans maybe to people, because we don't educate enough on mounds in general. So the fact is, we know that they weren't giant skeletons because we have archaeologists that dig burials up at these mound sites and we find that they are not giant. They are 5'6" to 5'10". Every single one of these books, this is a fraction of the archaeological record in just, the southeast mound sites. This is a fraction of the text that you can find in which every single one of these books detail burial records of burial practices, grave goods found with these Native Americans, everything from their height, weight, class in society at the time. We know it all, and we know they're not giant. So I don't know, people don't like academia, I guess, I'm not sure. The myth that goes along with this, that I heard a lot in parks, is beneath the whispers about giants, sometimes people would come to the visitor center and be like, hey, have you heard, about the lost tribe of the Israelites? Have you heard that Mormons built these mounds? And you can't really see it very well, but just know that this is a mess of maps overlaid on top of southeastern mound sites that say things like the Book of Mormon and the Plan of Salvation. They basically take mound sites and attribute them to their religion, their worldview, instead of, reading up or researching about a different people's worldview. This comes from something called the Jewish American Indian theory, and it's basically this theory that has prevailed since the 15th century, like 1500s, 1600s. White European explorers come through and they see a people that looks nothing like them, right? And they're like, that doesn't make sense. I believe in Christianity. Let me go back to the Bible. Let me see this scripture, and let me see if anything can be put together to make this make sense in my worldview. So they just thought that Native Americans were the lost tribe of Israelites that have populated out from the Bering Strait and Greenland. They came over and they populated out through North America. And then the Mormons took that and rolled with it, and this became such a problem at mound sites and parks that I worked at that, people of the Mormon religion would come through with the Book of Mormon and say, have you heard the great message that these mounds that you've studied your whole life actually were built by us? And I was like, oh, I have not heard. And I didn't really know what to say. So for about a year working at the park, I just didn't know how to deal with these people. And finally, I went out and I bought a copy of the Book of Mormon and put it under our front desk. And that fixed everything. I was like, hey, I already got it. And then the conversations would end. Instead of an hour or two of talking, it'd be fixed in 5 minutes, and they'd go on their way. So that is how I had to deal with that. And then the myth that I didn't see as much was, maybe I would get maybe 3 giants believers a day in parks for 5 years, and then, I'd get Mormons maybe every 2 weeks. But Vikings would be every few months. I'd get a person that came and they would think that the Vikings built the mounds. So this one makes a little bit more sense in the grand scheme of things. There is some physical evidence. They had mounds in their homeland, right? They came over and they do have a confirmed settlement in North America, but it is in Canada. It's in Newfoundland, technically. And this was in 1012. The settlement, the only settlement, by the way, that has been archaeologically confirmed from Vikings in North America is from 1012. And just from my experience, it was 650 AD or CE when mounds at Toltec were being constructed. So this already doesn't make sense in our timeline. Vikings could not have gone all the way to North America, built all of the mounds that we see dated before they even arrived here. So this also doesn't make sense. But the reason this is such a big deal is that this stone, this little stone, I think it's called the Rock Creek Tablet, was shown to me about a year into parks, and it was a hoax that is still believed sometimes and is still referenced in these YouTube videos where Norse characters were found on this rock that was apparently found in a West Virginian mound site. And we now know that somebody planted it there, and they had gone to a 1752 textbook of Norse characters, and they inscribed it on the stone themselves and planted it in the mound site so that their mound site could make more money. So it's just things like that that get to become myths and legends and hoax at mound sites, and that's what I had to deal with all the time. So I hope you think that a little bit more in depth about mound sites now, and I hope that if you have any questions about the truth and Native American mound sites and all of these, accomplishments that Native Americans actually did, you come talk to me after the main talk. You can come ask me questions. It's not my job every day to talk about this anymore, so I'd love to talk about it with you guys. So, thank you.
Thank you, Melissa. There we go. Our main talk today is going to be done by Brad Ericson. Brad is a former professor and loves to talk. Warned us when he came in that if we offered him the time, he would do it, and he was true about it, and really has been interesting. Before we get to that, volunteer for a role. While you're here, we're going to have 5 people presenting things today. You can do one of those 5 things. It's not that difficult, and we don't laugh at you unless you want us to. But the free thought, the member story, the skeptical sidebar, hosting, all of that's open. And maybe you have a speech you'd like to give. So volunteer for a role. See somebody here with a name tag on and say, this is what I can do, and I want to do it in a month or so. I think next month we're full, but the next month is available. The Arkansas Society of Freethinkers is a diverse organization with members who represent a wide range of opinions. We invite speakers who we think will be interesting and make us think. Emphasis on the make us think. Today our featured speaker is Brad Ericson, and Brad's talk is entitled Creation Arguments from Genetics. When you have times, please, when it's time, if you have questions, please wait till we get to you with a microphone so all the viewers at home are able to hear. And here now, Professor Brad Ericson.
Thanks for that introduction, Bern. Before I get into the meat of the talk, why do you suppose creationists attempt to use genetics to make their points? Any ideas? Why would they do this?
Verification.
Sorry?
Verification.
Verification, okay. Verification of what? What are they looking for? I mean, you're on the right track. What are they looking for? Sorry?
Of their mythology.
Of their mythology, okay. And in particular, what timeline are we talking about? What timeline are we talking about? Anyone want?
10,000 years?
So, yeah, 6,000 years, okay? So that is why the arguments that I'm going to critique today have been put forth, okay? They are attempting to legitimize a 6,000-year-old Earth through genetics, okay? So that's the idea. So I started off here with a quote from Albert Einstein, which I think is very apropos today, both from what we just heard as well as what you're going to hear. Okay, so those that seek knowledge should be congratulated, but unfortunately there's a number of people who misuse this approach. Now, do we have any Calvin and Hobbes fans in here? Okay, so you know what Calvin Ball is? Okay, so Calvin Ball is a game that Calvin and Hobbes play all the time, with the rules being very fluid. As soon as Calvin starts losing, the rules seem to change. Okay, and as we're going to see, this happens with creationist arguments all the time. Okay, is this? Yeah, there we go. Okay, so I'm going to present 5 main arguments that creationists go through to attempt to make their case. First of all, they will say that junk DNA doesn't exist. And again, I'll be explaining what is meant by this term here in a little bit. Second, random sequences cannot generate genetic information. Third, a new concept called genetic entropy, which I think you'll find rather interesting. Then created heterozygosity, and then finally continuous environmental tracking. Okay, so here's our main players. So from the Discovery Institute, Stephen Meyer, Casey Luskin, and Michael Behe. You probably heard at least of Michael Behe, but maybe not the other two. From Creation Ministries International, John Sanford. From Answers in Genesis, Nathaniel Jeanson. From the Institution for Creation Research, Randy Galuza. And here's a group photo of them getting ready to go to a conference on creationism. Okay. All right, so argument one, and this is primarily from the Discovery Institute. Information only comes from intelligence. Okay, so creationists will argue that information can only come from design. Okay, we've got no random components, only purposeful construction. And again, as I indicated, these are primarily Discovery Institute arguments with the intelligent design approach. However, there's plenty of evidence to the contrary. And the first is junk DNA. Now, when the human genome was completed, when the sequencing was completed, it became very clear that only 2% of the human genome actually contains genes. Okay, that was rather surprising. I remember prior to it being sequenced, people thought there was going to be about 100,000 genes in the human genome. It turns out there's on the order of between 20 and 22,000. Okay, depending on exactly how you characterize a gene. Okay, so much, much less than expected. So what does that mean? That means that a huge amount of the human genome does not specify proteins. Now, just because it doesn't specify proteins doesn't necessarily mean it has no function, and that's what the creationists are going to argue. Okay, and in some degree they're correct, because we have to have sequences that control those genes. We have to have sequences that specify RNAs only, that is, they don't actually become proteins later. Okay, so we're well aware of sequences that are not involved in producing proteins or functioning as genes. Okay, but even with that, only about 10 to 12% of the genome is actually utilized. And we refer to this, the reason we know this is because only that percentage of the genome is under constraint. Okay? In other words, when we examine the human populations as wholes, okay, what we find in common is, sequence-wise, is that 10 to 12%. The remainder, okay, is where the huge amount of variation takes place. Okay? So if you are ever confronted with the argument that all DNA has function, simply ask them, okay, what function can you ascribe to a piece of DNA that the sequence is not important? Okay? Because that is in essence what they are arguing. Okay? They will say that every sequence is important, yet when we examine the population as a whole, okay, we find that about 80%, 80 to 85% or so of the sequence has no constraint. In other words, the sequence varies between all of us. Okay? Okay, so purifying selection is the reason sequences are constrained. Okay, and so hopefully I'm able to explain this well. So first of all, looking at these two graphs, okay, we have fitness on the y-axis versus sequence variation on the x-axis. And on this panel, on the left panel, we've got purifying selection taking place. And on the right-hand panel, there's no selection. So when we have selection, okay, we're going to have the majority of the individuals in a population being around the peak of the curve. Okay? Now, why is this? It's because that if we have, oops, I hope I didn't change the slide. It's because when we have mutations that decrease fitness, what happens to those individuals? They reproduce less if they're less fit. That's literally what we mean by fitness, those individuals that are able to reproduce effectively. Okay? If you reproduce less, what happens to those genes? Those genes start to disappear out of the population. Okay? That's what we mean by purifying selection. Okay? That can occur on either side of the curve. We can result, oh darn it. Go back, there we go. We can have mutations that decrease variation, or we can have mutations that increase variation. Okay? Either way can result in a decrease in fitness. Okay? Whereas when we have unconstrained sequences, okay, that is sequences that have no consequence if you have a mutation within them, what we see is a flat fitness curve. Okay? So there's no cost from the mutations in these sequences, whereas we have a fitness cost in these particular sequences that are important to gene function.
What do you mean fitness? Like physical-wise, body-wise?
Right, so again, fitness is a measure of reproductive success.
How fine?
Okay, it becomes rather obvious in terms of animal populations, for example. Now, in human populations, it's because of modern medicine, okay, fitness effects aren't as stark as they used to be, okay? But with animal populations, you're going to have a rather quick decrease of less fit individuals. Okay? And of course, that means their genes will also be less. Okay. Now, in addition, you can take huge deletions out of a mouse's genome and produce viable mice. Okay? Why would this be? Well, there are regions within the mouse genome that are called gene deserts. That is, those regions of DNA have no specific genes within them, have no functional genes. We can delete those sequences completely, and the mice are perfectly fine. Okay? So that is direct experimental evidence, okay, that shows that we have these non-functional stretches of DNA within genomes. Second, can random sequences be shown to have function potential? And the answer is yes. Okay, creationists would reply no. Okay, random sequences they would expect to not have function. Okay, so here's the experimental approach. They synthesized 150 base long oligonucleotides. So remember that the genetic code is read in triplets. Okay, so one triplet specifies one amino acid. So if we have a 150 base long stretch, okay, that would have the potential to code for 50 amino acids. Okay, 150 divided by 3. All right? Now, what do we mean by, doggone it, I keep pressing on my, what do we mean by synthesizing something randomly? Well, we simply allow either an A, C, G, or T to be at every position along that 150 oligonucleotide stretch. Okay, so totally random, okay? No pre-design, shall we say, to that oligonucleotide. So we have this huge pool of 150 bases long oligonucleotides that have completely random sequences. So if we take those particular oligonucleotides and we express them in E. coli cells, we get a rather amazing result. Up to 25% of the random sequences actually enhanced growth, and 52% actually caused inhibition of growth. So 77% of the random 150 nucleotide stretches of DNA that were placed into these E. coli cells had a function. Okay? That would seem pretty good evidence against random sequences not being able to do something. Now, can random sequences be shown to have function potential? So in order to understand this, I'm going to have to go through a little bit of the lac operon. Okay, so the lac operon was the very first regulatory sequence that was ever described, is described by Jacob and Monod in 1961. And what this is a gene control circuit in E. coli bacteria. And there are three main components to it. So a regulatory gene that produces a protein called a repressor, and then the RNA polymerase promoter. So remember, we've talked about promoters before. So promoters are where we get the molecule known as RNA polymerase to bind, and RNA polymerase is what's going to result in the expression of these so-called structural genes. And then the third major component is what's called an operator sequence, and this is where the repressor binds. So the concept is relatively straightforward. If a repressor functions as a roadblock, if you will, so we got this big sign, road closed, okay, the RNA polymerase comes in, it can't proceed past the block when the repressor is there, okay? But when we have what's called the inducer, and in this case it's the actual sugar that is metabolized by one of these structural genes, when we have the inducer, the repressor is released, and now RNA polymerase can proceed on down the road, if you will, okay, and start making the mRNA for these structural genes. Okay? So, once again, we're going to take random sequences, this time only 103 bases long, because that is the stretch of DNA that we're replacing right here. Okay, so we've removed the wild type lac promoter, and we're substituting for it these random sequences of 103 bases in length. We've also removed the lac repressor gene. Okay, so all we have are random sequences that we are testing to see if they'll act as promoters. Okay, so the bottom line, and I'm not going to go through all the details here, okay, but the bottom line to this is a routine scan for lactose utilizing colonies produce colonies. Okay, so why would that be? Well, that means that these random sequences have function. Okay, they're able to substitute for the promoter. Okay, drive the expression of the E. coli genes, and as a result, we're able to detect those being able to grow on lactose as a carbon source. So this LAC-Z gene is what allows the E. coli cells to utilize that sugar. Okay, so if this gene was not expressed, you would not get any colonies, but it clearly is. Now, how successful was this? About 60% of the random sequences required only a single mutational event to become a functional promoter, and 10% of those sequences did not require any evolution. In other words, they were able to function directly. Once again, random sequences, no problem. Okay? were able to generate function. Final example with random sequences is, can we generate functional molecules directly? Okay, and in 1989, Tom Cech won the Nobel Prize for his discovery that some RNA molecules act as enzymes. Prior to this was the domain of proteins only. Okay? Proteins were the only molecules thought to be able to function as enzymes. But Tom Cech was able to show that this is not the case. RNA molecules have this property as well. And this is what led to the concept of the RNA world, which maybe I'll talk about sometime in the future. Now, the most important ribozyme that we all have and every living creature out there that you see has is a ribozyme known as peptidyltransferase. And without this particular enzyme, which is an RNA component of the machinery that makes proteins in your cell, without that enzyme, life would not exist. Okay, so ribozymes are extremely important. Okay? And I'm not going to go through all the details here, but realize that once again we started with random RNA pools. We allowed some laboratory evolution to take place, and they were able to produce, or identify, I should say, a 45 nucleotide long ribozyme. Now that's pretty short. Okay, a 45 nucleotide ribozyme, that's pretty short. And the ribozyme activity was sufficient to carry out synthesis of itself and its template. Now, once you have a self-replicating molecule, what do you now have the potential to do? Sorry? Okay, well, replicate, true. Okay, but what else? What's the key? If you have a self-replicating molecule, what can you now do?
Build something more.
Build something more through the E word, evolution. Okay, so once you have a self-replicating molecule, now you can evolve. Okay? And so, once again, random sequences easily can be shown to lead to functionality, which can lead to, of course, evolution. Okay, so can anyone think of what creationists may say to attempt to argue against this evidence of random sequences, of these experiments? Yeah, but they were designed. Scientists designed these experiments. Okay, that's literally the argument that I shouldn't say we, but that the scientific community has been hearing back. Well, of course it worked. You designed it that way to work.
Like cause and effect, you know, see the effect of something, you look at observation. Scientists observe the effects from certain situations, right?
I'm sorry, what now?
Scientists like observing certain effects of certain situations, right? Just observing the effect of certain, like scientists' observation of effects from.
Well, yeah, obviously the experimental observations were made, sure.
Right.
Yeah, so yes. Okay, so bottom line to this series of experiments and observations, junk DNA does indeed exist and comprises the majority of the DNA in animal and plant genomes. And generating the information for genes, the motifs for gene expression or self-replicating molecules are indeed well within natural processes and do not require intelligence. Okay, so on to some newer arguments. So genetic entropy. Now, genetic entropy refers to the gradual accumulation of harmful mutations in a population's genetic material over time. The origin of this proposition was put forward by John Sanford, and he has an entire book out there describing this approach, shall we say. The mechanism, he posits that as mutations occur, they can lead to decreased fitness and adaptability of organisms. And the implications is that genetic entropy may challenge the long-term viability of species, potentially leading to extinction. Now, think about that last sentence, or that last point, okay? What is he attempting to show or attempting to posit?
That life would be unstable unless there was some supernatural hand keeping it.
That's one aspect to it. The other aspect is mutation rate. So if we see a particular mutation rate, and this is true, then we can't possibly be around longer than 6,000 years or we'd all be extinct. Okay? So that's where this is leading to, okay? So what is entropy? Some of you may not actually have a good feel for that. So entropy is simply a measure of the disorder of a system. And the simplest way to think of it is think about water. So water as ice has the least amount of entropy. As it melts to water, that entropy increases, motion of the molecules increases, randomness increases, and as it goes to steam or a gas, okay, we maximize entropy of the system. Okay, the molecules are in the greatest degree of disorder. Now, genetic entropy is a made-up term. Okay, it was invented by creationists to describe a concept that already existed, error catastrophe. So the term genetic entropy is an attempt at branding. And you'll see this over and over and over again with creationists attempting to assign something as a brand. They can make a process seem more dangerous or inevitable through changing the name. Now, it should be noted that error catastrophe has never been observed or documented in nature or experimentally. Now, to conclusively demonstrate error catastrophe, you would have to show two things. One, that harmful mutations accumulate in populations over generations. Not just a couple individuals, has to accumulate in populations. And second, that these mutations cause a terminal decline in fitness, meaning that they cause the average reproductive output to fall below 1, meaning the population is shrinking and will ultimately go extinct. So genetic entropy supposedly works like this. Mutations that are only a little bit harmful, dubbed very slightly deleterious mutations, or VSDMs, occur, and because they are only a teensy bit bad, they cannot be selected out of the population. So they accumulate, and at some point they build up to the point where they are harmful, and at that point it's too late. Everybody's dead. Okay, so it reaches a point where the fitness is too low and the population can no longer reproduce. So there are at least 4 problems with this model. It considers only lethal or deleterious mutations. Neutral mutations are allowed but ignored, and beneficial mutations are not even allowed. Maximum fitness is assigned to the starting mutation-free genotype, so any mutation that elevates fitness is excluded, and compensatory mutations that reverse the effect of deleterious mutations, i.e. beneficial, are also not allowed.
So he's basically assuming away selection.
Go ahead.
He has basically assumed that selection cannot take place.
Correct. So his evidence for this is that he claims that previous generations of humans were smarter. Okay? That's why when we reached the moon, we found the ark on the moon. But of course, this is only a claim, and he has no evidence to back up this claim. Okay, but we do have evidence for this not being applicable to higher organisms, such as mice. Okay, and this paper just came out this year. Okay, and the general approach was as follows. So they took a donor mouse, and what this mouse is going to donate is a single somatic nucleus. So we just take it, say, from a liver cell or a spleen cell, what have you. Okay, so anything but a germ cell. Okay, anything but a germ cell. And we place this into an egg cell that has had its nucleus removed. Okay, so the only genetic material being provided is from this donor mouse. Okay? And we allow this to continue out for quite a few generations. Okay? And we simply ask, does viability eventually go away? Okay? Do we eventually lose the ability to clone serially these mice? Okay? So everyone understand the experiment? Okay? So donor mouse, and then clone 1, and then clone 2, and we're just doing the same process over and over again. Okay? So let's take a look at the results. Okay, so this experiment began in 2005. So note that this took some patience. Okay, this was a 20-year-long experiment. Okay, and you're looking at the success rate of cloning, okay, versus the generation. Okay, so success rate on the y-axis, generation on the x-axis. And here's the years as the experiment went along. So what does this mean? So this means that, for example, if we had 100 eggs that we attempted to provide a nucleus to, okay, out of those 100, we had about 7 successful clones. Okay, and then 5 and 5, etc. Okay, about 12 here, 14 here. And at generation 25, we peaked at about 15% success. Okay? But once we reach generation 25, look what happens. Okay? We start having a rather rapid decline. And finally, at generation 58, okay, we reach extinction. Okay? The clones are no longer viable. Uh-oh, is Jeanson right? Let's take a look. Or Sanford, excuse me, is Sanford right? Well, no. And for the simple reason that this is not how mice reproduce. Mice don't reproduce this way. Okay? Note that we always use somatic cell nuclei. We never use germ cell nuclei. Now, why is that such a huge deal? Okay? And the reason is because of a little thing called genetic recombination. Okay? The huge value of sexual reproduction is that we're able to undergo recombination during meiosis, separating mutations one from another. And so as a result, you produce viable gametes versus those gametes that would not be that desirable, okay, may even be lethal. Okay, and so as a result, you separate good sequences, if you will, from the bad. Okay. So two factors appear central to the collapse of the long-term clonal propagation. We get emergence of these large structural variants. So we get large regions being deleted or inverted, lots of mutations that are greater than 1,000 bases in length. These are rarely seen in the germline. Okay, so by doing this serial propagation, we generated these types of mutational aspects. And then, as we already mentioned, we get absence of chromosomal recombination when we do serial cloning. Okay. Now, additional points falsifying genetic entropy. Selection is very efficient at the phenotypic level, despite alleles being effectively neutral. It does not take much at all to shift, remember our fitness curve, okay, to shift fitness one way or the other. Okay? i.e., those alleles will not be the favored ones to be maintained in the population. Okay? And populations harboring the most genetic diversity should have the greatest decline in fitness according to genetic entropy. However, we see the exact opposite. We like to have diversified populations. What happens when we have inbreeding? That's when we get mutations accumulating to a lethal point, right? Okay.
Is there Roman time, things like that, like inbreeding or the children, like reptiles, and you're inbreeding, that's why it affects their genetic development?
Well, it would. I mean, it would affect it, yes.
Even their mind, that's why often paraplegic, they develop psychological and neurological disorders and all kinds of.
Yeah. I mean, all you have to do. I mean, we've witnessed what happens with inbreeding, in human populations, right? We've witnessed that.
Rothschilds are inbreed, that's wrong when they do a lot of. They don't go outside their bloodline and do it. They like stay within their bloodline. That's why they behave neurological disorders, like a lot in certain families for purity.
Yeah, I mean, mental retardation is certainly one aspect of inbreeding. Yeah. Okay, and so this illustrates the concept of crossing over. Okay, so during meiosis 1, we get pairing of the homologous pairs of chromosomes. We have what's called a chiasma form, okay, between two regions of the homologous chromosomes, resulting in exchange of those two regions. Okay, and so again, this is a extremely important aspect to maintaining, okay, genetic diversity and eliminating mutational load during reproduction. Now, there was a famous geneticist, Mueller, who predicted what would happen in the experiment we just discussed. And it's called Mueller's ratchet. Okay, and so he proposed in 1964 that harmful mutations evolving in asexual individuals will be passed to all offspring. Harmful mutations will accumulate across generations. However, only half of the offspring would get deleterious mutations in sexual species. Now again, if you happen to be the unlucky individual that gets the harmful mutations, what's happened to your fitness? Gone down, right? Okay? You're going to most likely either not be able to mate at all or mate very rarely, and those alleles will be eliminated, okay, will be selected against. So sexual reproduction reduces or limits the spread of deleterious mutations in the gene pool. Okay, well, cross off genetic entropy onto created heterozygosity. Now this is a fun one. Now let's remember what we mean by heterozygous versus homozygous. Okay, so if we're looking at a chromosome pair, okay, so in this particular chromosome we have both of these alleles being, shall we say, the dominant form, and here, okay, we have both alleles being the recessive form. Okay, this is what we mean by being homozygous. Okay, both loci have the identical gene sequence. Whereas if something is heterozygous, okay, we're going to have one of these alleles, okay, and one of these alleles on each chromosome. Okay, so that's what we mean by being heterozygous. Now we just described that heterozygosity is what we typically desire. We want variation within the population, okay, provides a healthy aspect to the genetics of that population, okay? So, what's being proposed is that this heterozygosity was created in the garden, right? So we've got every organism that at that time, 6,000 years ago, was fully heterozygous and happy as could be, right? Okay. So this is a claim popularized by the creationist Nathaniel Jeanson. And again, he's at Answers in Genesis, which is Ken Ham's organization in Kentucky, which I'll be there protesting it on July 12th, if anyone wants to join. But anyway, so from the previous slide, okay, realize that some genes have hundreds to even thousands of different alleles. So let's jump back. Okay, so envision, for example, there being 100 to 1,000 different variations of this particular gene. That's what we're talking about in the population, right? In an individual, they can have only one of them. Okay, one form of the allele. But in the population, we can have a huge number of different alleles. Okay. Now, question. How could all of this variation arise in approximately 6,000 years from creation, or even more quickly, 4,000 years since the flood? Okay. So according to created heterozygosity, Adam and Eve were created with every gene in the heterozygous state, as were all other organisms. Now, note that they have rapidly left the realm of science here. Okay? This is obviously a non-falsifiable statement. Okay? Any statement you make in science must be falsifiable to qualify as science. So how do you get the variance? Well, there's a number of ways that we are well aware of that can generate genetic diversity. So one we've already discussed, genetic recombination, and mutations, transpositions. We talked about transpositions a few weeks ago. And gene conversion, which is another process which I'm not going to go into. According to Jeanson, there's also some divine involvement in these processes, i.e. tinkering. Okay, so once again, okay, note that they've now left the realm of science by admitting this. Okay, so remember the second premise on which science is based. All observable phenomena are the result of a physical or chemical cause that is measurable. Okay, no miracles allowed. It also contradicts three laws of science. Must be testable against the natural world, must be explained by reference to the natural world, and again, we have to frame things as if so that they can be falsified. And you don't get to just make stuff up and call it science. And that's exactly what has been done here. Creationists are just assuming an unobserved, ongoing divine intervention. Jeanson claims his model, which he put forward in 2017, predicted the nested hierarchies that we observe today. And just in case you needed a little reminder what we mean by a nested hierarchy, so here's an example of a nested hierarchy for the vertebrates. Now, this is not true. Created heterozygosity is not observable, and his predictions are not predictions, as we already knew about these hierarchies well before 2017. So taxonomy, remember, should reflect evolutionary relationships. And a good way to show this is the box in a box concept. Okay, so here's our various taxons, so species, genus, family, and order. And I just stopped at order. I didn't continue on. Okay, so if we have, and note that the larger the taxon, the more it includes, right? Okay, so order is all of this. Okay, and then family is the green boxes. Okay, so within an order, we can have multiple families. Okay? And then within families, we have genuses. Okay, so the white boxes. Okay, and then finally we have the individual species. Okay, and what this taxonomic ordering does is provide an evolutionary tree. Okay, it shows us who is related to whom most closely. But the biggest problem for this model, the maximum number of alleles in two individuals is 4 for any given gene. Okay? So just quickly, okay, so for any given gene, okay, we can have 2 differences, right? So that's all that we have possible. So, for example, let's suppose that chromosome 9 has big A1, little a1 in individual 1, and big A2, little a2 in individual 2. So there are human genes with way more than 4 alleles. And the best example is the ABO blood group. It has greater than 1,000 different alleles. Okay. Now, to understand this problem, we have to describe the difference between intergenic recombination and intragenic combination. Okay, so on the left-hand panel, we have intragenic combination, that is, within a gene. Okay, and this is what I was talking about earlier, how purification works. Okay, so if we have 2 mutant genes within a particular individual, and we undergo recombination at the correct location within, okay, these 2 genes, we'll end up with a double mutant in one particular gamete and wild-type sequence, that is, without mutation, in the other gamete. Okay, so again, this is why recombination is so important to maintaining genetic purity. Okay. So again, this is intragenic recombination. Here's intergenic recombination. Okay, so between genes. And this is the example that we looked at earlier. Okay, so we get crossing over at various points. Okay, and we simply exchange genes between chromosomes. We don't do anything to the genes themselves. Okay. Jeanson is talking about recombination between genes. But his problem is, to generate all those different alleles, we need recombination within genes. Okay, if we're going to change the actual sequence. So recombination within genes is much rarer than recombination between genes. And remember, as we discussed earlier, the protein component in the human genome represents only about 1% to 2% of the genome. And in addition, remember in eukaryotes, genes are usually split into the portions that express the protein and those that are in between the expressed regions, i.e. exons and introns. And so we're going to have recombination within the exons only. Okay? Within the introns won't help us. Okay? And intra-exonic recombination is rarer still. So the math for the number of alleles shows why Jeanson arguments are preposterous. So using Jeanson's numbers, now remember he's got this book out there, okay, and he gives these numbers. So using Jeanson's number, he starts with a 15-year generation time. Now, in humans, while this is not biologically impossible, okay, it's probably a bit unlikely. But we'll be gracious and let him have his 15-year generation time. Now, this is the number I could not figure out where he got. I wish I would have emailed him and asked him where he got this number from. But anyway, we'll let him have it. So he claims there's 40,000 recombinations per lineage. Okay? So remember what we mean by lineage. So a single breeding pair. Okay? So no one, his wife, and his 3 sons and their wives would be a lineage, each a lineage. Okay? So that means that there's approximately 870 recombination events per chromosome. Okay, so 40,000, again, that's his number, divided by 46 total chromosomes. But again, we'll be generous and we'll round this up to 1,000. Okay. Now, if we assume equal distribution of recombination events across chromosomes, which is incorrect, okay, you do not get every chromosome equally crossing over, and equal likelihood within chromosomes, which is also wrong. There are regions within chromosomes that undergo much more recombination than others. But again, we'll let him have these assumptions. Okay, so to generate the greater than 1,000 ABO variants, the ABO gene is 25,000 bases in length, or 25 kilobases in length. It's located on chromosome 9. And chromosome 9 itself is about 140 million bases long. So if we divide 25,000 by 140 million, okay, we get 1/5,600th of the chromosome represented by this particular gene, okay, that designates our ABO blood groups. Now, if we assume each recombination generates 2 new alleles, we would therefore need 500 recombination events to get our 1,000 alleles, okay, within the ABO gene, starting with 4 alleles. Okay, so again, okay, Noah has 2, his wife has 2. Okay. So assuming all recombination locations are likely, this requires 500, okay, times, okay, the fraction of length of the ABO gene in chromosome 9, which means we need 2.8 million recombination events in chromosome 9. Okay. So at approximately 1,000 events per chromosome per lineage, again, remember this is from Jensen, that requires 2,800 distinct lineages radiating from the arc. But Jensen's problem is he's only got 4. Noah and his wife, his 3 sons and their wives. Okay? So we're a little short of 2,800 lineages. So now let's look at the real numbers, not Jensen's. Okay, so there's approximately 1 crossover per chromosome per generation. So again, we get the same 2,800,000 generations needed. Okay, and again, we're only going to get 1 crossover per generation. So at 15 years per generation and 6,000 years, okay, we need 400 generations per lineage. Okay. And this means we would need 7,000 lineages. Again, we've only got 4. But also, recombination, as I pointed out, is not equally distributed. Most occurs in less than 10% of the genome. This makes the problem worse by a factor of 10 to 100. So the conclusion is that we cannot generate enough diversity in the young Earth creationist timeline of 6,000 years, even with the extremely generous assumptions that we allowed Jensen to have. We would require, with created heterozygosity, 500,000 years. So we seem to be missing 494,000 years somewhere. Okay, so created heterozygosity, cross that one off. So continuous environmental tracking. Now, this goes back to an interesting question that was asked in the early days of molecular biology. How do mutations arise in organisms? Are they spontaneous or are they induced by their environment? So, as I indicated, this was a fundamental question back in the early days of genetic research. And in fact, it goes back even farther than this. This takes us all the way back to Lamarckism versus natural selection. Remember that Lamarck believed that new features are the results of environmental conditions, whereas Darwin realized that different organisms arise and develop through natural selection of small inherited variations. And in 1943, a seminal experiment was performed by Salvador Laurier and Max Delbruck, both Nobel Prize winners, by the way, called the fluctuation test. So let's describe the fluctuation test. If bacteria are resistant to virus, okay, they will not be lysed. Wild-type bacteria are sensitive to the phage and will be lysed. So we start with cultures of wild-type bacteria, and we could observe one of two possible outcomes. The first possibility is that the mutation was pre-originated. Mutations are already present in cells that provide the resistance, and they're random. That is, we have probabilistic mutation. The second possibility is that bacteria exposure to virus triggered mutation. The resistance is driven by the environment. Only became resistant upon virus contact, that is, the organism is responding to its environment, and mutations do not happen randomly. So here's the experiment. So each of these represents a separate culture. So we started with a small number of bacterial cells. We allowed them to grow in culture. We then plated them onto nutrient agar plates that contain virus. Okay? So once again, remember that if the cells are sensitive, they'll be lysed. If they're resistant, they'll form colonies. Okay? They will not be lysed. Okay? So if we have induced mutation, here's what we would expect. We would expect, okay, about the same number of resistant colonies regardless of the culture, right? Okay? So if the mutation is induced, okay, by the presence of virus, then we would expect about the same number every time of resistant colonies to arise. However, if we have spontaneous mutation, we're going to expect diverse results. Okay? So in this case, for example, a single mutation occurred late that provided resistance. Here, a mutation occurred earlier that provides resistance, and therefore we got more resistant colonies. Here we had no mutations that resulted in resistance. And here we had a mutation that occurred in this generation and generated resistant colonies from that lineage. Okay, so note several things from these results. So if the mutations were induced, we would expect a consistent mutation rate, okay, and that's what's being shown here. Okay. Each individual culture, however, showed a different result, that is, random mutation. And the lineage for each mutation can be followed. So, for example, in this one, okay, we could follow that lineage and identify where that initial mutation occurred. And if the mutation occurs early in the culture, this leads to more resistant cells. And this is, again, the result that Luria and Delbruck attained. Okay? And remember that this was in 1943. Now, I'm not real good at math, but 2026 minus 1943 would be about 83 years in my estimation that we've known this. Okay, so here are some quotes from the ICR, or Institute for Creation Research website. Our task is to fundamentally change the way people understand biology by constructing a completely new theory of biological design that incorporates recent discoveries and, note the recent discoveries of only 83 years ago, and respects the biblical narrative, all with the goal of igniting a second creationist revival. Continuous environmental tracking is a new model that flows from the latest findings in molecular biology. Again, only 83 years behind. Identifying innate sensing systems and logic mechanisms that direct targeted responses to environmental challenges. Living creatures are active, problem-solving entities that continuously track changing environmental conditions and through innate systems purposely adapt themselves over time to better fit existing niches or fill new ones. ICR is at the cutting edge of developing an approach to biology that's totally current, fully pro-life, got to have that in there, right? And truly Christ-honoring. Our goal is to build a clear, concise, and easily communicable biological framework that explains biological functions from an organism-centered, engineering-based perspective that gives glory to the Creator, not to nature. With the scientific insight and enablement of the Lord Jesus, we are moving forward to seize this extraordinary opportunity. Now, if that isn't as anti-science as you can get of a description, I don't know what is. So this is what happens when you start with a conclusion and try to find data to support it, as opposed to following the data where it leads. It also demonstrates a total lack of understanding biological mechanisms that have been described for centuries in the case of Lamarckism versus natural selection, and 83 years in the case of the Delbruck-Lurie experiment. This would seem to contradict the cutting-edge claim for continuous environmental tracking. All right, so let's wrap up. So does information only come from intelligence? No. There's no known nucleic acid sequences that function without maintaining sequence specificity. And again, at most, we have 12 to 15% of the human genome that is constrained, i.e. functional. Plus, we would not be able to delete huge portions of a genome and maintain a functional organism if this were true. Random sequences clearly can have function, and we showed this for proteins and RNAs. We showed this for gene expression motifs and for ribozymes. Genetic entropy is simply a made-up term for which there is no evidence. And the experimental evidence is all to the contrary. So Phage T7 mutational load produced actually improved fitness clones. And mouse serial cloning showed the importance of meiotic crossing over and demonstrated the ratchet principle proposed by Mueller and does not support genetic entropy. Continuous environmental tracking is an outdated idea that goes all the way back to Lamarckism and acquired characteristics. And the Lurie-Delbruck fluctuation test conclusively demonstrates the random nature mutation and disproves continuous environmental tracking. Continuous environmental tracking created heterozygosity and genetic entropy all demonstrate what happens when you attempt to try to make the data fit your preconceived conclusions. And in every case, current scientific data is either ignored or misrepresented. Thanks so much.
Are you familiar with the term life force, like life energy, life force, spiritual energy?
Now, the concept of vitalism, which is in essence what you're...
Like spiritual energy.
Right, right.
I mean, because allegedly, we're all surrounded by energy field. Even plants, they can measure some energy, rate energy of some kind. Like spiritual force, life force, how does that affect evolution? Because I believe the concept that, a life form mutates to its environment, like it changes based on the fit the environment it grows in, like the life force, spirit, energy that animates.
Okay, so I'm going to address that. Okay, so the concept of vitalism, which is in essence what you're referring to, concept of vitalism was debunked, and it was done by Craig Venter's group when he synthetically, 100% chemicals, okay, purely synthetically made the genome of a bacterium and placed that genome into a bacterial cell that had been enucleated, had its genetic material removed. What happens? We transform that particular bacterial cell into the cell whose genomic material was now placed in it. Okay? 100% synthetic. Nothing about that required any magic, any life forces, anything like that. Okay? Vitalism was put to bed by Craig Venter. Yeah.
I have a question. Back to, I think it was like slide 3 where you had that bell curve of variability and fitness.
Yeah.
And to understand that graph, I understand it as the whole population's variability and the whole population's fitness in terms of, likeliness to reproduce in a given environment.
Yes.
So is it accurate to view that graph on the left there as variation decreases, that is something similar to inbreeding? Like if you inbreed dogs, for example, you will eventually get such low variation that they don't reproduce very well in a given environment.
Yes.
On the other hand, if they're in the middle and they're well diversified, they'll breed really rapidly. And then on the other hand, if they're put in the presence of mutagens, for example, and get a lot of variation, then they would reproduce more infrequently because they're all getting exposed to negative mutations. Would that be an accurate?
Yes.
Okay.
Yep, that would be correct. Yep. Now again, the reason this doesn't happen, okay, I'm going to ask the audience a question here. So what is the reason that doesn't happen in populations? What did we just describe or spend time describing today that prevents that from happening?
Mixing and matching.
Mixing and matching, okay, so genetic recombination, okay, followed by selection of alleles, natural selection of those alleles, okay, and remember that natural selection is extremely good at identifying phenotypes that are optimal versus those that are not. Okay, and so that's why we cluster around the peak and we don't see sliding fitness. Okay. That it? Chris, you got some more?
Yeah, so it seems like the two sides in this debate are operating by different rules, and that those rules...
Calvin Ball!
Like the rules of science inevitably lead to this, right? Because science, cannot allow for untestable hypotheses, appeals to, supernaturalism, or there was another one. There's the rules of science that you have to follow, or else you're not practicing science. The faith-based system, has a different set of rules. Namely, you can't, question the dogma, or you can't. You have to make things fit the dogma rather than questioning everything. And I wonder, to what extent does, we can talk about the evidence that you've presented a lot of. But to some extent, it's people's different definitions of what is a fallacy. Like, in the scientific world, an untestable hypothesis is like a logical fallacy. In the religious world, it's just like, day-to-day operation. This is what we do.
Sure.
So I wonder, even going down the path of experimentally testing things in science is interesting because we're using science and the rules of science to debunk the creationist argument in terms of science, which they don't acknowledge using their method, which is, arguably pre-Socratic, I guess you could say. The evidence doesn't matter. What matters is the fitting it activity and your role in doing that. So I don't know. I guess is there ever a point where all the research fails to change minds because they're using a different philosophy, and we need to back up to the philosophical level?
Yeah, so in the Dover PA trial, Kitzmiller v. Dover, it was argued by the defense, okay, i.e. the intelligent design folks, that, well, scientists are going to miss out by not allowing the supernatural to be considered part of science. They're going to be missing out. They're going to be missing all these great new hypotheses that could be tested. And the simple answer to that is, how would you test them? To describe to me how you're going to test whether or not a supernatural entity is carrying out natural functions. How are we going to distinguish that? How are we going to test for it? That is the straightforward answer to that argument, okay, that response. And, if you haven't studied Kitzmiller v. Dover, do it. I mean, it is extremely enlightening.
I'm curious, what was their answer to that?
They didn't have one. They did not have an answer to how they could test for it. They said, well, we're working on it. I think that was literally what they said. We're working on it.
Recently.
Well, that goes back to they have a different approach to epistemology, like where does knowledge come from? They would not even think of having a testable hypothesis because that's not part of the rules they're operating under.
Right, yeah. That's exactly right.
Even researchers can be influenced by their beliefs and things like that.
Of course. So why?
As an idea, you can't evaluate or change your views based on new information or what's going on with it.
Right, and in science we change our views all the time. That's the beauty of it. Okay? Whereas dogmatic thinking, if you start with Genesis 1:1 and that's your starting point, where can you go from there? I mean, anything that would contradict it now must be discarded or ignored.
Plus, they justify a lot of atrocities in their own time. The Crusades, there's a lot of people that religion or belief justify faith or cruelty against other people or even genocide.
Of course.
Your faith.
Yeah.
Could you explain again, I missed something in that talk, what exactly did you mean by tracking?
Okay, so in other words, the reason we see adaptability in nature is because the organism itself is able to identify its environmental conditions it's in and adjust gene expression accordingly. But as the fluctuation test demonstrates very clearly, that is not at all what happens. Okay? We do not have organisms able to identify a bacteriophage, for example, and suddenly develop resistance to it. Okay, that is not what happens. Okay, so that's what is meant by tracking. Okay, they're literally identifying where they are in an environment. So, this is something I thought of in terms of another test. Okay, well, let's take a plant from the tropics and move it to the Arctic and see how it does. I think I know what's going to happen. I don't think there's going to be any environmental tracking. I think it's going to die and die very quickly. Okay? So that would be another very simple thing to do.
Unless you get enough of them. Unless you get enough of them.
Unless what?
You get enough of them.
Go get enough of them? Yeah, I suppose if you...
if you get enough of them, one of those will survive.
Well, no, I would argue that it wouldn't matter how many you put up there, okay, you're not going to get survival. Okay, none of those plants are going to survive.
You're being dogmatic.
Now, I suppose it might depend on what you mean by survival, because we're well aware of organisms that have been frozen for millennia that can be revived. Okay, and we're seeing that happening as unfortunately the Alaskan tundra, for example, melts away. We're seeing organisms that have not been alive for thousands of years, maybe hundreds of thousands. I don't think they've identified any millions yet, but I know at least a number of bacteria that they've been able to isolate have not been around for 30, 40, 50,000 years.
That's why cryogenics and stuff like that. I think in cryogenics is that as well.
Right.
Cryogenics is free. I think in Jurassic Park was that the theme where they had like a gene from a mosquito or something like that with a basis for like.
Yeah, that was right. That was the presumed basis. Now, interesting. We can't have Jurassic Park, but we could have Pleistocene Park. Okay, we could have Pleistocene Park because we have mammoth genomes that we've been able to sequence in their entirety, and so the so-called approach would be to take that genetic information, place it in an elephant egg, you allow that egg to develop to term, and then interbreed them, and eventually you would get very close to an authentic mammoth. But anyway.
Just by chance, I think we've had a synthesis of between the skeptical side of our presentation and this one. She's encountering people who are talking about these giants of the past that may have, built the mounds, and then the genetic entropy folks are talking about a genetically perfect humanity of Adam and Eve, slowly degrading over time and into becoming a non-viable species, I guess, at the end times in their religion or whatever. And so there's a crossover there, I think, between the genetic entropy folks and the people talking about ancient giants. That maybe the ancient giants were the genetically better than us organisms, and we are the degraded versions of what they used to be,
Yeah, I mean, that's in essence what they, in some instances are arguing. That people in the past were genetically superior, and we're degenerating and much less intelligent. And so, like I said, that's why we found the Ark on the moon. So yes, of course, that's where it was. Should have known that's where they would hide it. Go ahead.
Well, it could be that the discovery institute and the other related folks are the evidence of the decline.
I think that's a pretty good argument, actually.
I might go out and live here and stuff like that, and maybe it's considered or something like that, but a lot of black projects, there's a lot of stuff they don't tell us. There are so many hidden involvements or advancements and things like that.
Mm-hmm.
What do you think there's a point where they can actually clone a whole human body? I mean, I think they can clone humans now.
Well, I mean, that is a worrisome but very real possibility, human cloning.
Because allegedly, Blaine Maxwell, which was Jeffrey Maxwell's old friend, Senator Jeffrey Maxwell, she gave testimonial that they actually they were underground abducting children or individuals, and they have underground facilities where they're actually cloning whole people and stuff.
Wait a minute, that's what killed Hillary's campaign. I don't buy it.
Well, likely they can't clone humans. They say that's it, but they break down, like you say, they burn out quickly because there's no.
Right, now you couldn't serially clone, but with chimpanzees, they have cloned a chimpanzee. And as I've described a couple times, we're about a half a chromosome away from being chimpanzees ourselves, right? So yeah, it's definitely possible. I just hope that bridge doesn't get crossed. I would really hate to see that. All right, thanks. Sure.
Thank you, Brad. Now what we need is for Brad to come up with a very simple, simplistic, explain it to me like I'm 5 description of how the mRNA vaccines work. Because that
I've given a talk on that before.
okay, here?
Not here.
Then work on that. But do it like the Reddit, the subreddit says, explain it to me like I'm 5.
Right.
Get to that. Okay, well, hope you've enjoyed today's meeting. Here are our announcements. You can see our full schedule at meetup.com, but book club is next week at Pocket Dimension Bookshop at 2 o'clock?
Yes.
2 o'clock. Pint Night is always the 3rd Monday of each month at Flying Saucer downtown Little Rock at 6:30? 7. That time has changed a lot over the years. Sometimes it was my fault. First Tuesday is always the first Tuesday of each month at 5:30 at American Pie Pizza in North Little Rock near Kroger. Second Sunday Cantina Communion is the second Sunday of each month at Cantina Cinco de Mayo on Highway 5, and that is at 6:30?
6 o'clock.
6 o'clock. That's Jim's, he protects that. The Arkansas Society of Free Thinkers needs volunteers. We need people to help with graphic design and website development, to write letters to stop church state violations, and to host events like potlucks and family and child-centered activities. If you have skills or ideas to contribute to our cause, contact us at info@freethinkers.org, or better yet, just talk to somebody here and say what you can do and what you'd like to do. Get involved and come to some of our other events. Our calendar, again, is on meetup.com. On the table, you will find the Celestial Teapot, where you can feel free to leave a donation. In the meantime, feel free to stop by, have a snack, and talk with some people. Thank you for coming.